This is Dr. C, recording from home. I've missed everybody. Studio's been closed now for over 6 weeks. And unfortunately, those of us who have regular programs were unable to go up to the studio. So we'll do it at home. Apropos to the times now, I want to talk a little bit about, first, the immune system in our body. A lot of people tell you what's good for the immune system, what's bad for the immune system. What do you really know about the immune system? Well, first of all, let me try to put this all in lay language for you. After we talk about the immune system, we'll talk a little bit about the testing that's been done, or being done now, not being done, I should say, too, in Massachusetts, and we'll talk a little bit about vaccines. So let's get started. First of all, what the heck is the immune system? The immune system itself will recognize self, the cells that are in your own body, and non-self, anything that would invade the body. So, very simple terms. It recognizes self and non-self. Then that sets up a whole series of different things that will happen and steps to try to get rid of the non-self. First of all, let's talk about the self. Our bodies turn over their cells regularly. Depending on which organ we're talking about, they turn over at different rates. In other words, maybe a year from now, you'd be looking at a whole different person because basically all of my cells have turned over. It happens. It happens. You don't even realize it. And talk a little bit about the two different types of things that are going on with our immune system. First of all, plants, all the other animals all have an immune system. It's not unique to humans, although we do have parts that are unique to any mammal that is able to chew, which means us, basically. And so, first of all, what's common to plants and lower animals? Well, first of all, they may have an external coating on them, which helps to protect them from external type of organisms. Could be just simple things like a waxy substance on their surface. Could be in many different things. Could be a particular odor that will keep and repel insects or even animals from them. Now, also plants may have stuff that will attract certain insects, like bees, for example, to pollinate them. So this is going on in both plants and in humans as well. That type of system is also in humans. It's an innate system, it's called, an innate system. We're born with that system. It's innate. In other words, when you're born, your mother can pass through the placenta, pass that particular immunity to you. And you're going to use that for a period of time till you develop your own. And it may take maybe 2-3 months, and you've got your own system then. And so, that particular thing is passed on. So, let's go from there. What's the first thing that happens when your body recognizes non-self? Now, this could be, say, a genetic problem. Let's say you get a mutation of a gene, takes off, becomes what we would know as a cancer. Or let's say your body got invaded by a particular bacteria, or nowadays, a particular virus. What happens then? First thing that happens is that your body will start to have increased flow of what they call lymph. Now, lymph is just a distillate of your blood. Take away all the cells. That's what they do in the laboratory when you go to get your blood test. They spin it down, get all the cells out, and then they use the serum in order to do whatever test they got to do. And so the lymph system then produces additional lymph, which goes to the area that got invaded by this particular non-self entity. That will also carry with it certain messengers now that are going to activate the system, the innate system now, the innate system. What the innate system does, first of all, there are certain white blood cells that will produce immunoglobulins called Ig. You'll see this term, big I, little g, and then there's different immunoglobulins in that system. We can get those measured in the laboratory. They're doing that all the time, measuring the different immunoglobulins for different reasons. So, first of all, the B cells, these lymphocyte cells, B cells, will be producing these immunoglobulins. Now, the immunoglobulins themselves will be produced at a different rate, and they come on at different times. The first immunoglobulin to be produced is immunoglobulin M, IgM. That's a big molecule, pretty good sized molecule. Usually starts on about the third day after identifying this, what they call a pathogen or their non-self. About the third day to about the 12th day, right in that range. After that, your body will produce IgG, big G, IgG. That's the majority of the immunoglobulin that we have in our body. That has more lasting power. That's the one that you should have measured and see if you have immunity to something, IgG. There are other immunoglobulins as well. IgA, for example, which is in the secretions in your, say, nose or saliva or whatever, all the secretions will have some IgA. We'll see that IgA, for example, in people with a lot of allergies, for example. You'll see IgA. You also may see IgE. IgE is a very small amount compared to the other immunoglobulins. In other words, it's about probably the rarest of all the immunoglobulins. And it comes out usually produced by what's called mast cells, M-A-S-T, mast cells, related to histamine. Mast cells produce histamine. And you've probably heard that term, especially if you have allergies, because that's why we take antihistamines to try to fight the allergy. So these are the different types of IGs that are produced, immunoglobulins, and they float around in our serum in our body. So we have that in common with a lot of the other animal kingdom as well. What else besides surface or besides immunoglobulins? Well, now we also have what's called an adaptive system in our body. This is true now for all chewing animals, including us, of course. We still chew. Okay. So we all have this adaptive system. That's mostly cellular. That's cellular. Let me talk a little bit about the white blood cells in our body. There are many different kinds of white blood cells. The most common white blood cell out there is a leukocyte. Leukocyte. The leukocytes are part of the innate system, that system that is in our body and gets passed on. Innate system. What happens after our body produces those immunoglobulins, that stimulates also production of these leukocytes. And then that stimulates the leukocytes to go to the area that is involved with this foreign material, non-self. Each leukocyte can actually then engulf and eat, you might say, eat up to 15 or so virus particles or bacteria or whatever. And that kills them. It lyses them. Also, the leukocytes could be part of going after cancer cells as well, because that's non-self as well. So the innate system basically is immunoglobulins and leukocytes. Now you notice that if you have an infection, for example, they do a white blood cell count. And they look at the number of leukocytes. Well, of course, if you have an infection or you got some invasive thing, your leukocyte count will go up because your body now in the bone marrow is producing these leukocytes to go to that area of inflammation or infection. Now let's again talk about that adaptive system. The adaptive system is mostly the large round cells in the white cell population, the round cells. They are called lymphocytes. There are several types of lymphocytes. They look a little different when you look at the morphology, look at the anatomy of them when they stain them. Look a little different, but they have their particular function. The two biggest types of lymphocytes, basically, are B lymphocytes. Remember I told you B lymphocytes produce those immunoglobulins. And also T cell lymphocytes. These are the killer type cells. These are the ones that will go after any invading bacteria, virus, whatever it is. The T cell or killer lymphocytes. Now our body has a whole system of things that go out into your serum that will notify those lymphocytes that this is going on, and they'll produce, your body will produce more of them, and they will all go and migrate to that area of either inflammation or infection or whatever. So you've got B cell and T cell. The other part that's true in the humans as well is they have memory cells. Again, they are lymphocytes, or what we call macrophages. Macrophages are large round cells compared to the smaller lymphocytes. The macrophages are of two types. They could be floating around in the serum as well, or they could be fixed in particular organs. And so when the blood comes through that organ, if the macrocyte has been focused on getting rid of a particular invasive organism or whatever, it will grab it as it comes through. It's a macrophage. And so you've got the whole system of lymphocytes and macrophages as part of what we call now the adaptive immune system. Okay, very good. So, knowing that as the basic stuff now for our own immune system, what can we do to help and what can we do to hurt our own immune system? Well, let me go through some of the things. How do we, first of all, strengthen our immune system? Well, first of all, you could go do a lot of exercise, regular exercise. Simple thing, won't cost you a cent. Walking, simple exercise. Remember, 150 minutes a week. of exercise. Number 2, get enough sleep. That strengthens your immune system as well. Number 3, vitamin D or sunlight. One way, either getting out, getting the sunlight, or taking vitamin D in, one way or another. But vitamin D, adequate amounts, strengthens the immune system as well. Now there's dietitians and nutritionists out there who want to sell you everything. And there's a whole variety of things that they will tell you are good for your immune system. And there's no question that it may be, especially in a test tube. Are they good for you anyway to spend a lot of money? Well, you got to be that judge. Things like elderberries, mushrooms, acai plants, oysters, watermelon, low-fat yogurt, dark greens like spinach that also have folate and fiber, tea, especially green tea, sweet potatoes, broccoli. All of these different things are supposedly helpful to promote your immune system. On the other hand, how do you weaken your immune system? Well, first of all, age. You get to be an old fart like me, it's going to be a lot less strength in my immune system than somebody who's 20 years old. I admit it. But we can still strengthen it. How do we weaken it? Just the opposite of the things I told you. Don't get enough exercise. Be a couch potato. Number 1. Number 2, lack of sleep. Some people think they can get 2-3 hours a day and that's it. Nonsense. You need that sleep. Number 3, chronic anxiety. If you're worried about everything and everybody, it's now chronic anxiety. That not only will keep you from sleeping, but it also is not good for immune system. Low vitamin D. Some medications are also weaken your system. A big example of that are steroids. I feel sorry for the people who have asthma, for example, who have to be on chronic steroid use all the time. I feel badly for them because their immune system is definitely going to feel it. People who smoke, diminishes their immune system. People who have diabetes, weakened immune system as well. And so all of these things add up. You want to try to do the best you can to try to improve your immune system, of course. And so you just follow those basic guidelines. You don't have to spend a lot of money. You don't have to go and buy a whole bunch of different food additives, etc. Just eat your regular diet, keep your weight steady, exercise, get enough sleep. Okay, so that is the immune system. Now let's talk a little bit about what we're going through right now, the COVID-19. I wanted to especially dwell on that a little bit. First of all, the testing. If you've paid attention at all to what's in the media now, the people who know the least about virology, especially virology, because that's what the COVID-19 is. It's a virus. The people who know the least about it are our politicians and the news media. That doesn't mean you can't listen, because they'll give you proper statistics and all, which I do listen to, the statistics. They may not be very good, but nevertheless, the statistics are out there. But don't go any further believing anything else that they're telling you. Most specifically, why don't we go through some of the tests that are being done What are good tests? What are bad tests? What do we need for tests? Well, first of all, the most common test that's done out there, which is using that swab, that nasopharyngeal swab. One of the big things that I first learned about some of these so-called tests that are out there that tell you, first of all, that you have the virus. Well, the first test that was approved, basically, was this one that still is in use. And I wanted to refer to it. I just have to find my notes here, and I apologize for that. Here we go. It's called the PCR test. Actually, RT, real-time PCR. Who cares what it stands for? It stands for polymerase chain reaction. Does that mean anything to most of us? No. But basically what they do, this test, it identifies a portion of the RNA gene. RNA is ribonucleic acid. That's part of the gene that's been identified in the COVID virus. That test has been available. And it's got a pretty good sensitivity. Sensitivity means what means that you have a very low false positive rate, which is good. It's very sensitive. The problem with that particular test, one of the problems with that particular test, is that there's a fairly substantial false negative rate. That's called specificity, false negatives. Now in China, it was up to 70% that were okay, 30% false negative. What does that mean? That means if you actually had the virus, 30% of the Chinese were not picked up by that test. Now, of course, in the United States, when this came out, the companies that were producing this, and that included our own FDA, included them as well. I don't exonerate them from anything. They were claiming 95% specificity, which was baloney. Baloney. It's closer to about maybe 80 to 85% specificity. How come you get these false negatives? Remember, false negative is when you have the disease and the test doesn't pick it up. It's negative. Well, first of all, error. Error. Maybe the person doing the test didn't actually get that swab all the way in and rotate it a little and pull it out. Not an easy test, because you got to go all the way back to the nasopharynx. A lot of the tests were just swabbing a little bit of the snot. That's it. Some of the tests that came back to the lab were dry. You couldn't even analyze them. And then some of the tests, you didn't have enough reagent. Remember when the test came out, our CDC said, no, we can't use the World Health Organization tests. We're going to do our own. And what the heck did they do? After a delay of 2 weeks, they put it out, sent a bunch of them out, and believe it or not, the reagent didn't work. Finally, they got their act together and sent them out. And this is the test that's being done to tell you if you have evidence of that RNA gene, in other words, the COVID gene, in your nasopharynx. Now, why did they choose nasopharynx? Well, first of all, COVID is a virus that first infects from respiratory droplets, gets in your nose, goes to the back of the nose. You get a sore throat. Then it goes down. How does it get into the cells? Well, research has shown that it gets into the cells because on the surface of the cells in our own respiratory tract, we all have it, on the surface, there's called an ACE, ACE2 receptor, ACE receptor. The further down into the lungs and the further down you go, the more of these receptors are present. The virus, the COVID virus, uses that receptor to get in. How does it do it? Because one of the proteins on the surface of that COVID virus, right on the surface, attaches to that receptor and the virus gets into the cell. The only way the virus is going to cause all sorts of havoc in a body, it has to get into the cells. That's the only way it can replicate. It has to get into a cell. If it can't get into a cell, can't replicate, can't cause all sorts of destruction. So this is how it gets in, and that's why the lungs are really the central organ system that is initially infected by COVID. So this test done properly will tell you, if it's positive, that you have the virus. So that's number one, the PCR test. Still around, still being done, and that's the one that they do to tell you if you have the virus or not. So if you're negative, does that mean that you will never get the virus? Of course not. Remember, there is a significant false negative rate. And just because you don't have the virus one day doesn't mean you don't have it the next day. So you have to not only do the test, but you're probably going to have to repeat that test several times over the course of time. So, PCR. Now, with our labs here in Massachusetts and all, they came up with one of the tests that check the antibodies. Now, what are antibodies? Remember I told you about the immunoglobulins. They are forms of an antibody. They're an antibody. Especially that IgG, which is our main source of immunoglobulins. And so, if that test that they do will pick up the IgG, they will tell you that you have antibodies towards coronavirus. Does that mean you have COVID-19? No, because there are a number of coronaviruses. In fact, there are 7 of them that are known to be around in the circulation, and people will get, say, a common cold from the coronavirus. No more than that. There are 4 coronaviruses that cause these common colds. This coronavirus test, this antibody test, doesn't necessarily tell you you have COVID-19. It'll tell you if you have been exposed to a coronavirus. So, again, it's not specific. The only value to the test is that if you at some point in time were positive with that first test, that nasopharyngeal test, and now you have a positive antibody test, now you can correlate the two and say, okay, you probably had COVID-19 and you're getting an antibody towards it. Let's go a little bit further. The value to the test that was produced and now is being used, the Abbott test. Not specific for COVID-19, but it does tell you it's against a coronavirus. That test, the value of it basically is that it's quick. Within 5 to 10 minutes, they can tell you if you're positive. If you're negative, does that mean you don't have coronavirus? No, it doesn't. Just tell you have not had time to produce the IgG, which usually doesn't start for maybe 10 to 14 days after you've had the infection. And so that's why that test is of value, and also the drawbacks to the test. You can't make broad statements about it. So that's a second type of test that's out there. They talk about, too, these lateral flow assays. Now, some of you may have heard of a lateral flow assay. For example, if a young lady worries that she's pregnant, she puts a drop of urine onto that strip and then reads those little lines that come out, whether she's positive or negative for pregnancy. That's a lateral flow test. The urine goes that way. Now, what they've done is isolated those genes or those proteins on the coronavirus. Isolated them. And they also now have isolated something that will stick to that particular gene, couples with it. So when that lateral flow occurs, when you put some blood or whatever onto that strip, as it goes along, that it will hit that stuff that's going to bind to the particular protein of the coronavirus, and you will get that color change. That's a lateral flow test. A little bit better than some of the other tests, but still does not tell you if you have immunity at all, and may or may not be specific for coronavirus. Remember, there's hundreds of tests out there. Only 4 tests have been approved by the CDC. 4. So keep that in mind when you get the test. You can go one step further, actually 2 steps further. You can get what's called an enzyme-linked test, an assay, enzyme-linked assay. This is called an ELISA, E-L-I-S-A, ELISA test. That goes a little bit further. What that does, basically, it will quantify either IgM or IgG or both. Takes about 4 to 5 hours, however, to run that particular test, and it has to be done in a laboratory. So how you do that is you have a plate, and on that plate, you coat it with a viral protein. If you happen to pick a protein from COVID-19, and there are several diagnostic proteins on a COVID. Now, you remember what a COVID, God, they put it up on the TV dozens of times a day. You got these little spikes coming out, little spikes. Looks like pieces of rice coming out of a meatball, for example, just these little spikes. Well, that spike protein is what a lot of them are using now on that plate, a spike protein, so that when you put the serum onto the plate, you can identify and quantify the amount of IgG and IgM that you're producing. People who produce a lot of IgG, at least in theory, have a better chance at immunity. Not guaranteed. Nobody will say it's guaranteed at all. But at least it quantifies the amount that you have. So that's another type of test that's done. An interesting type of test that's on the horizon uses genetic engineering. Now that is really fascinating to me, this genetic engineering. What they can do now is they can actually take that RNA. They've identified the genes that are on the COVID virus, single-strand RNA. They've identified the genes. And they can actually change the gene. They can take that particular gene off and either keep it off or substitute a neutral gene for it. Very interesting way of doing it. In fact, the labs that are doing some of this, it's called a CRISPR technique, they may have something for the future in terms of vaccine. So for now, those are the tests that are available. The types of tests can alter what your treatment plan will be. And certainly, you don't know how many people out there are immune, because our government, our own CDC, right from the start, knew we didn't have enough tests. And so they say only if you have symptoms should you have the test. Baloney. The countries that got better faster did them on everybody. South Korea, for example, did them on everybody. Yes, they need to produce a lot more tests. Where's the holdup? Where are the tests? I don't know. All these millions of tests that are supposed to be out there, you can't necessarily find them, and you can't necessarily just go in and say, I want a test. I don't have any symptoms. I want a test. How can you tell if a population out there has what's called herd immunity? Enough people out there to prevent an epidemic. How do you know? How do you know there's enough people out there who can stop an epidemic because there's enough people with immunity? The only way is to test everybody, get a percentage. If you have 60% or more of the people out there who have antibodies to that particular virus, you have herd immunity. That's another term that is used. Not explained to you usually, but that is what it means. So that is the COVID testing regimen. Let me tell you a little bit about vaccines. I know a little bit about vaccines because we have produced vaccines for a number of years against a lot of different diseases that are out there. And I ran the St. Anne's Free Medical Program for 20 years, and we gave out vaccines. Do you know the difference between a vaccination and immunization? A vaccination is just giving you an injection with a vaccine. A vaccine is an antigen. It's going to stimulate an antibody response in your body. That's all it is. The other thing, too, about vaccines versus immunization. Immunization is when your body produces the antibody, produces an immune response to the antigen that you got in the vaccine. Just like getting the disease. Produces an antibody, and that antibody may or may not last a long time. There are several types of vaccines, and obviously they're trying to produce a vaccine against COVID-19. There are two major types of vaccines: live attenuated vaccine and inactivated vaccine. Now, if you take a virus, how do you inactivate it? You can do that either by heat or chemically. They usually use formalin, and that inactivates the infectious part of that virus. Some of the vaccines that they produce now will use just an inactivated vaccine. On the other hand, you can have a live, attenuated virus. What does it mean to be attenuated? What they've done is they've taken that virus, identified what is the infectious part of that virus, and remove it from the vaccine, remove it from the virus, or do something to alter it so it no longer will infect. So that's the two types of vaccines that are out there. The inactive type of vaccine doesn't have longevity. An example of that would be the tetanus shot that you get. You said you got to get it every 7 to 10 years. Why? Because you begin to lose immunity as time goes on. That's the nature of the inactive type of vaccine. Many of the vaccines now are live attenuated. Attenuated, not totally live, they're not infecting you, but attenuated so that they don't cause infection, but they can still promote the antibody production against that virus. So most of them now, like for example, hepatitis B. Hepatitis B, you have to get 3 shots in order to get and build up immunity, at least to the point where you're not going to get any active infection from the hep B. How long does that last? We're not sure. We're not sure. A lot of times it appears that your immunity is going down, but then if you do get or confronted by that infection, all of a sudden you are able then, because of the memory cells. Remember I told you you also have memory cells in those lymphocytes. You also have memory cells that will pick it up and go through again the type of sequence that happens to promote these antibodies to form. Okay, so those are the two types of vaccines that are out there. And companies that are trying to produce these vaccines may use one or the other type, live attenuated or inactive stuff. What can they do to promote antibodies? Okay, let's specifically talk about COVID virus. There are proteins on the shell of that virus. Remember, there are little spikes, and that spike protein is one of the proteins that will promote antibody formation. There are other types as well. The envelope that covers the virus, called an E protein, that may be enough to stimulate the production of antibodies. There could be an M protein, the membrane, or it could be an N protein, the nucleocapsid, N protein. Most of the places that are trying to develop these vaccines use either the spike protein, the S protein, or the N protein, because they can stimulate antibody production in greater quantities and faster than the other proteins. So those are the ones that they're trying to produce. Very interesting, the latest on the vaccine production now for COVID-19 seems to be coming from Oxford, England. They were working on a vaccine for the MERS, the Mid-East virus, which went through here back in 2010, 2012, in that range. And they were working on the vaccine towards that virus. MERS is a coronavirus, and it has a lot of proteins that are in common with the COVID virus, COVID-19. COVID-19 is more of a SARS virus. That came through back in 2002, 2004, right in that range. And so and that is a coronavirus as well. But the present one is closer to the SARS than the MERS. But at any rate, getting back to the Oxford laboratory, they were working hard at trying to get a vaccine for the MERS virus. And so when this epidemic came around, they now are trying to produce the vaccine now for the COVID-19. Now, is that good or is it bad? Well, at least it gave them an upper hand on how to produce the virus, and they got things a lot quicker. They're in the labs, and they got things a lot quicker. They got a big chunk of money now from AstraZeneca, and they should hopefully get it online, producing millions of vaccine doses by the end of 2020. We're hoping. So that's basically what the latest is on that. Now, the COVID-19 is a very interesting virus. It's estimated that maybe 70 to 80% of people who are exposed to it will get very minimal, mild, or even no symptoms. That doesn't mean that they don't have the virus, and that doesn't mean they can't spread the virus. Usually between 2 to 3 to about roughly 12 to 13 days, you can expel and be actively spreading the virus, even though you have no symptoms. That's why it's so important to test everybody, despite what the CDC says, despite what they say. Like I said, don't always listen to everything, because number one, they don't have enough tests, and so they make excuses why you shouldn't, if you don't have symptoms, why you shouldn't get the test, which is total baloney. Total baloney. We should all get that test, all of us. So, at any rate, it's an interesting disease to have. You may, for a while, not have a lot of symptoms, and then suddenly, if that virus gets into the lower portion of your lung, remember I told you it goes through those ACE2 receptors and gets into the cells, replicates rapidly from that point. All sorts of little maneuvers that our doctors will use in the intensive care units, and they, by and large, it's a learning curve. They have to learn how to do it. But they are doing the best they can, given the amount of equipment they have and the lack of protective equipment that, again, the CDC keeps saying there's plenty out there, you just have to, baloney. All of the states had to compete against each other to try to get enough of that equipment. And they're competing against the federal government. So even a state like Massachusetts or New Hampshire, if you finally get an order in for, say, 1 million masks or 1 million whatever you want to get for protective equipment, once that protective equipment gets to your facility, most of them in the past have been stolen outright. Stolen by our federal government, FEMA. They don't have to ask. They just come in and take it. Where does it go from there? Supposedly it goes to our national stockpile. That's another interesting thing, the national stockpile. The stockpile consists of about 12 different areas around our country, warehouses, where they store essential equipment in case of an emergency. They also store medicines. Not only vaccines and personal protective equipment and all that other stuff, like ventilators, things like that. They also store medications. I have a whole talk about the medications and how our wonderful, wonderful government deals with how to preserve the medicines that they have in that national stockpile. You don't know where these 12 places are around the country. They won't tell you that. They won't tell you how much of everything is in those national stockpiles either. How come people aren't asking about this? How come people don't ask about the medications that they put in this? Do you know that the federal government asks every year for the FDA to run some tests and see if that medication is still okay, even though the expiration date is not? Over 80 to 85% of all the medications that are in there get approved for maybe up to 10 years. 10 years on that national stockpile. Can you get your medication looked at? No. Not even any private organization can. A state can't even. So there's a lot of things that are kind of rotten in Denmark. So I think I've talked to you enough today. I want you to take home at least a few of the pointers that I made out. First of all, the immune system, how it works with the COVID and their testing of the COVID, and testing and vaccine production for COVID-19. I would hope to talk to you soon and get you a little bit more information. And I'd like to have more of these sessions, not only about COVID, but as we did before, issues, especially issues that are pertinent to seniors. I want to talk to you about a lot of different things, and I've had plenty of time now to get a lot of these things together. I want to produce more exercise programs for you as well, and I would hope to do this hopefully in the near future. Thank you, and we'll see you shortly.